post-fertilization structures and events. So post means after. Fertilization means the fusion of male and female gamete to form a zygote. So what happens after the fertilization is called as post-fertilization. So we have the pre-war, the war, and the post-war, right? In the same way, in case of fertilization, we have the pre-fertilization, the double fertilization, and the post-fertilization. And so now we are here, the post-fertilization. So, following double fertilization, there are some events of post fertilization, and I just mainly divided them into two events. So, first we have the events of endosperm and embryo development, and second, maturation of ovule into seed and ovary into fruit. And these events are collectively termed as post fertilization events. So, first, let's learn in detail about the endosperm development. Endosperm development precedes embryo development. Why? It is because the primary endosperm cell, PEC, the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserved materials and are used for the nutrition of the developing embryo. So just remember this. The primary endosperm cell divides to form the triploid endosperm tissue, and this triploid endosperm tissue contains food which is required for the nutrition of the developing embryo, and that is why endosperm development precedes embryo development. And in the most common type of endosperm development, the primary endosperm nucleus undergoes successive nuclear divisions to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei form before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. So just remember this one. Coconut water is the free nuclear endosperm, and the white kernel is the cellular endosperm. One is free nuclear and one is cellular. An endosperm may either be completely consumed by the developing embryo for their nutrition. Okay, example maybe if the endosperm is consumed completely, then this type of examples we can give it as the pea, the groundnut, and the beans, and or endosperm may persist even in the mature seed. Example, we can see this in castor and coconut. See, we still see the endosperm, that is the cellular endosperm, and the free nuclear endosperm in coconut, right? So that means that the endosperm is not consumed completely by the developing embryo. So this is all about the endosperm development. Embryo. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed because we know that the embryo development is later than the endosperm development, right? Because they have to depend on the nutrition which is produced by the endosperm. So most zygotes, they will divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Okay, here is the answer. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. For example, you can see monocotyledons in corn in corn, and dicotyledons in beans. And figure 2.13 depicts the stages of embryogeny in a dicotyledonous embryo. Okay. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. So let's look it in the figure. So we say that the zygote will give rise to the number 1 proembryo, number 2 globular embryo, number 3 heart-shaped embryo, and number 4 mature embryo, right? As you can see from this figure, this is our zygote, right? So this zygote will give rise to the proembryo, and again next the globular embryo. So this looks like a globe, right? Globe, globe. So it is called as globular embryo. And if you can see from here, this looks like a heart, right? The heart shape. It doesn't look like a heart actually in terms of biology, but you know, it looks like a heart. Just remember it as that way. And this is the heart shaped embryo, and this is the mature embryo. And now these two coming out will elongate, okay, and this two will become the cotyledons, and this is the radicle, also called as the root tip, okay. And this part you see here, right, this part, the ending part, this is called as the plumule. So just remember it now, right? So the zygote will give rise to number 1 proembryo, number 2 globular embryo, number 3 heart-shaped embryo, and number 4 the mature embryo. Atypical dicotyledonous embryo, which we can see in figure 2.14a, consists of an embryonal axis and 2 cotyledons. So the portion of embryonal axis above the level of cotyledons is the epicotyl, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyl, that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. So let's look at the figure. This is the epicotyl, okay, the embryonal axis, and these two are the 2 cotyledons, and this is the hypocotyl, and the epicotyl and the plumule, they terminate each other. So epicotyl, and this is the plumule. The plumule is also known as the stem tip, and the radicle here, it is also called as the root tip, and the root tip or the radicle is covered up with a root cap, okay. And so from the name itself we can know, isn't it? So the plumule is the stem tip, so it will give rise to the stem, and the radicle, which is the root tip, will give rise to roots, okay. So just remember it in this way. Embryos of monocotyledons possess only one cotyledon. So we can know from the name itself, mono means one, cotyledon means the cotyledon, right? So one cotyledon. So embryos of monocotyledons possess only one cotyledon. And in the grass family, the cotyledon is called scutellum, that is situated towards one side of the embryonal axis. At its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated shed called coleorrhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyl. Epicotyl has a soot apex and a few leaf primordia enclosed in a hollow foliar structure, the coleoptile. So we're going to learn the difference between coleorrhiza and coleoptile afterwards. So let's look at the figure for now. So this is only for the grass family, okay. It is the only for the embryo of the grass, not for any other. So a grass is a monocotyledon, so it will have only one cotyledon, okay. And the cotyledon of the grass is called as scutellum, right? And the scutellum is nothing but the other name of the name of cotyledon in grass, okay. So this, it has only one cotyledon, and the cotyledon is called as scutellum. And these two, you see here, these two are the coleoptile. This is the soot apex, and this is the epiblast, and This is the radicle, which will form the roots, and this is the root cap, and this is the coleorhiza. Okay. Now let's see the differences between coleorhiza and coleoptile. Coleorhiza and coleoptile. Okay, so for coleorhiza, it protects the radicle or the root tip. Coleoptile protects the plumule or the stem tip. You can see here, okay, rhiza are root tip or radicle, plumule stem tip. So coleop, P, you see there P, right? Coleop, plumule or stem tip, and in the other side rhiza, R, radicle or root tip. So just remember it in this way. And since coleorhiza is related with the root, it will be towards the soil, and coleoptile, since it is related with the stem tip, it will be towards it will be upward direction from the soil. And coleorhiza does not turn green, and in the states you will not see the root turning green. In the same way, coleorhiza will not turn green at any stage, and coleoptile, you know, the stem is green, okay, sometimes, and the stem and then the leaves, you know, the upper part, so it will turn green color after emergence from the soil. And from this third point we can add another point, that is, if it does not turn green, coleorhiza will not photosynthesize, and coleoptile, since it turns green after emergence from the soil, it will photosynthesize. So we can write 4 points. I hope we are all clear with this coleorhiza and coleoptile.